Welcome for me today. We're very lucky to have Dr. Molly Hyer here, and she's going to talk about incorporating sex as a biological variable in research. She is a postdoc in Dr. Gretchen Molly's lab in anatomy and neurobiology.
Hi everyone, thanks for coming. I see some familiar faces in the lab, in my lab, but also in the room. So I am going to start off today with talking a little bit about my background and my own experience to kind of highlight why studying sex differences is pretty important to me and is an integral part of my research already. So these are my four primary mentors that I've had throughout my academic career. So Kelly Lambert was my undergraduate advisor at Randolph-Macon College. Dr. Owen Floody was my advisor at Boston University. Dr. Erica Buxton at Maryland. And of course, Dr. Gretchen Nye here at PCU in the Anatomy and Neurobiology department. So I've actually been studying sex differences with all of these individuals. So I've had males and females incorporated into my research. I started out in Kelly's lab looking at paternal and maternal behavior in mice and mouse species. So I do mostly animal models. So I'm going to be talking largely about that, but we did try to incorporate some aspects of human research as well, depending on how discussion goes. I started out looking at maternal behavior, and then for my master's degree in aggression and sexual behavior in hamsters. And then I kind of combined both of those for my Ph.D. with Erica, where I looked at sexual behavior and parenting. Those things go together if you guys know that. And then for my postdoc, I am working with Gretchen and I, where we are directly comparing males and females and how sex acts as a biological variable. So SHB is a common acronym that you guys may see as you are hearing more and more about sex differences as you are reading news and reading the articles as it is becoming more of a prominent research. So this is the outline of the talk I'm going to give today. So you'll see the bar along the top, and we'll kind of go, the letters will turn black depending on the section that we're in, so you can sort of follow along with where we are as we progress. We're going to go through some definitions of terms, sort of the scientific background of sex differences for the biological level and why and then we should care the National Institute of Health mandate about incorporating sex as a biological variable in your research applications. And strategies to address this mandate or just to address incorporating sex as a biological variable in general. And then finally, we'll sort of end with a little workshop where those of you who are here today can talk about your research and how you see sex differences affecting how you want to incorporate that into your life. So the first piece I think is very, very important to talk about up front is sex versus gender. So these two terms are often used very interchangeably. I want to be very specific here that today I'm talking about sex at the genetic level. So I'm talking about X and Y chromosomes and how they make a genetic male or female. So the definition for sex is male or female in a biological sense. Largely, non-human animals have a sex, so we can identify that very clearly. Gender, on the other hand, is male or female in a social sense. Of course, there can be some debate there, but for the purposes of this talk, I'm going to clearly state that. Non-human animals largely do not have a gender. So when we're talking about particularly animal models, and I'm talking about sex, I'm talking about genetically male or female. And of course, gender leads to gender roles, which are these culturally defined expectations that an individual is expected to fulfill. And then, of course, gender-specific roles are those that are based on gender and whatever societal constructs you have where you are living. So these terms can be oftentimes used interchangeably. So when you are talking about sex differences, when you are writing this into your grant applications and your papers, sex is the appropriate term if you're using animal models in this case. Gender can be very specifically defined if you're going to be using one term versus the other. Largely what I'm talking about today are sex differences. And so if there's questions about gender, we can talk about that maybe at the end. But of course, I do want to recognize that I'm going to point you in the direction of some resources. So obviously, sex and gender are quite a continuum, and the more research is coming out, we're even seeing a lot of evidence at the biomedical level of different gender identities. So I want to kind of recognize that And I'd like to point you in the direction of the work of Dr. Joanne Rogarten. So Dr. Rogarten is a trans woman who she's an evolutionary biologist, and she has been studying the role of sex and gender in human and non-human models for a couple of decades. Her work is somewhat controversial, but she is very engaging, and it is very interesting. So I can direct you to her book, Evolution Rainbow, if you are more interested in that. Additionally, the Organization for the Study of Sex and the International Society of Gender and Medicine are two societies that cover sex and gender in research, and they're wonderful conferences, very mentee friendly, very new person friendly. So those are excellent conferences to attend if you want to see what work is out there, or if you want to present work that you are doing on your own. They do everything from human research, animal models, pharmacology, behavior, genetics, the whole gamut. So those are both excellent conferences. So I've been quite a number of times. Okay, so what do I mean when I talk about sex differences? And let's kind of outline the definitions. So when most people think about sex differences, they think of phenotypic sex differences. They think of sex differences that you can see and that are very evident. And so we know that there are some very obvious sex differences. So of course, in spiders, you know that the female is almost 4 times larger than the male in any of the species. And humans look like distinct. Lions, of course, the male has the same, while the female does not. And the wood duck is a very good example of phenotypic sex differences where the male shows an extensive amount of plumage as part of mating rituals. However, many species don't have these evident phenotypic differences. And so hedgehogs, standard lab mice, and rats, although there are some size differences there, but many canine species, there's not overt sex differences between the males or the females. However, there's a lot of different types of sex differences. So as you guys are working to incorporate sex into your research, grant applications, all that stuff, these are some of the terms, again, like sex and gender, that you want to very carefully define. You want to very carefully use these when you're talking about your research. So there's a bunch of types of sex differences. So I can say sex differences, and we're like, oh yeah, male and female. But there's a more subtle distinction to be made depending on what your outcome is. Your data may be interpreted differently. So sexual dimorphism is what most people think about when they think about sex. So sexually dimorphic, with 2 distinct forms. This can be true both in appearance and behavior. So of course, the wood duck is a very sexually dimorphic animal, and they're sexually dimorphic right out of the egg. And as they develop, you start to see this plumage in males and not in females. Then we also have examples of different behaviors. So copulatory behavior is sexually dimorphic. Breaking displays are all regularly sexually dimorphic. So that means 2 distinct. But then, of course, we have sex differences. And so this is where the lines begin to blur a little bit. And so sex differences are what exist on a continuum. So there's a number of examples listed there, but I think a very easy example to digest is height. So in humans, men are more likely to be taller than women, but of course we know that this is a continuum as there is a large variety in height. So many men are shorter and many women are taller. So this is an example of a sex difference. So the averages are different, but there is of course a continuum. And then, of course, my personal favorite is sex convergence and divergence. So this is where it gets really exciting. So we see this phenotype is the same. So the outcome behavior is the same. But the biology is different. And so my personal example of this is the California mouse, Californicus. This is the species that I worked with for my Ph.D. studying parenting behavior. So this is a socially and sexually monogamous mouse. They bond for life. And the males are really, really good dads. The joke we always would make is that the males would mate if they could. They're very good dads, so they're a good model for parenting behavior. Those mice look exactly the same. You probably can't tell which one's male and female. And of course, they have different biologies for how they engage in maternal care. So what happens is, as the female is approaching birth, the male's testosterone levels start to drop and his estrogen levels rise. So at the moment of birth, the male's estrogen levels are actually matching the female's estrogen levels. But what they're doing at the mechanistic level is that the male's estrogen is driving him to stay in the nest, to stay home, because testosterone leads to more leaving the nest and exploring around, whereas estrogen is staying in the nest. And so you're having this different biological mechanism, whereas the female, of course, she's just gone through gestation and now birth. So her hormones are telling her to nurse, to groom, to nurse, to thermoregulate these babies, while the males are saying, "Oh, I got to sit here too." So their behaviors become the same. So this is the sex convergence. Opposite of that, of course, is a sex divergence. So a very common example of sex divergence is you see a lot of similarities in children prior to puberty. So a lot of their behavioral characteristics are the same. And then, of course, puberty happens and definitely diverge in conflicts. So those are two examples of convergence and divergence. So if you are incorporating sex differences into your work, you may fall into one of these different categories, and how you interpret that data will depend on how you write about that will kind of make your interpretation. And so by using these terms, you sound like you know what you're talking about. So if you want to have sex differences in your applications, incorporating these types of terms and at that subtle level of definition is going to really strengthen your interpretation. Also, you guys can ask questions. We're commentary and
So, these examples you mentioned later were necessary?
not necessarily. Largely, sex differences in any of these capacities are going to be hormone dependent, but not always. And so, I guess not a better example, but an additional example is that you can be direct or indirect relation hormones. Right? So, in both of the examples I gave, I talked about directly hormone dependent. There are other examples where your adolescent hormone state lays the foundation for your adult behavior, but that behavior could be dependent on LTP, LTD, which are not necessarily going to be hormone dependent. So you can have either direct or indirect effects. Okay. So why do we have these differences? And hopefully I don't have to say that too much to the folks in this room here, but of course we start out even before conception with sex differences. So these are just an image of the X and the Y chromosomes. And as you can see, the X chromosome is significantly larger than the Y chromosome. We're already starting off with a different amount of genetic material, even pre-conception. Then, of course, these differences only get exacerbated as we move through development. And so we're talking about hormones again here. But on the Y-axis, you see the gonadal hormone levels, or on the X-axis, you see different developmental stages. And in the males, you see blue sort of testosterone, pink is represented estrogen. Males and females have both. So males have estrogen, females have testosterone. Males just have a lot more testosterone, while females have a lot more estrogen most of the time. And those are going to drive a lot of these mechanisms. Is sort of what I was just trying to my answer to Drew's question shown in the graph. So this is from a neuroscience group, technical research was very helpful how to incorporate that work in your own and trying to move that field forward. But they're, of course, neuroscientists. So you'll see the examples down here are all neuroscience related pieces of how these sex differences test. So you'll see that the males prior to birth have a surge in testosterone that then sort of waves down as they approach kind of this juvenile phase, while females are pretty much baseline for that whole early developmental period. And then, of course, puberty hits and males just have an escalation in testosterone, while females begin the cyclical pattern of the menstrual cycle. This reflects in this pattern over development, reflects in these different aspects of neural development. So you see early on, already differences in apoptosis, neurogenesis, after pruning. So even pre-conception, which we're already starting to have differences in brain development that are going to develop. So it's completely sexual play during the juvenile period. So we have a lot going on across the world, and available. It's been a stage for a lot of these behaviors. And so depending on what time point you are looking at in your research, if you're looking at something like, I don't know, both if you're looking at age, animals or humans, these factors are all going to come into play. So a lot of people, so if you do cell culture work, you may think, oh, well, I do cell culture work. This is no problem. Well, you guys are taking cells, if you're using animal models, you're taking cells from early neonatal tissue, we already have sex differences in that neonatal tissue. So thinking about where you fall along this continuum and at what point your research is, at what point you're conducting your research is really important for how you interpret your sex differences. So here's, I guess, the meat of why we're even having this conversation. So this is, and I realize this is from 2011, but I did sort of specifically pick that date for setting up for the next slide. So this is just showing you, there's a lot of different fields listed over here, but just to kind of give you a global sense of how often males are reported in publications versus females versus both. And as you can see, there is a substantially amount of research focused on females. For reproduction, you see plenty of females represented, but that's obviously a very small piece of what females do throughout their lives. So despite these known sex differences, we have a dearth of research on females. And of course, harkening back to my earlier sex and gender, there's even more of a deficit in gender-related studies. So that is a caveat to take these different definitions. So what does this mean for research? Is this really that bad? So I could give lots of examples of this, but this is just one that I'm going to give that is very poignant and has been a huge topic of conversation for a long time now, and is only becoming more potent as it gets worse. So there is a considerable amount of evidence. So this is from this Nature paper published in 2019. So males and females have different pain thresholds. A lot of people, Drew, don't say, no, you can say whatever you want if I'm wrong. A lot of males so females actually have a higher threshold than males. And so a lot of people think that is the opposite, that males are going to display a higher pain threshold than females. That is kind of the stereotype. But when you look at the much of the research in this group did, I think they did the von Braun years, they show that females have a higher pain threshold. We know that there's a mechanistic difference for why this is the case. And so you see here the examples of how pain is modulated in males and females. But what does this mean for drug use? Well, females are prescribed opiates significantly more frequently than males are. So while we know that females have this lower pain threshold, we also are seeing this prescription increase in females compared to males. And so we have this sort of mismatch in how interpretation of the data is being used in medical practice. And so this is just one example. I could give a lot more about how not studying females leads to problems in actual medical practice. But this is just one, and of course, a very poignant one. So in 2015, the National Institute of Health posted this mandate. There has been a lot of pushing for this to come forward. And then in 2015, they finally did this. And so I've underlined sort of the relevant portion. So preclinical research studies continue to rely heavily on male animals and or omit reporting of the sex of animal subjects. This is particularly problematic in those studies intended to inform understanding of diseases and conditions affecting both sexes. So this mandate came out, and that meant that now sex differences became a very significant portion of current applications and research publications. And so the previous excuses are no longer tolerated. So I don't have enough space to have females in my colony. Not good enough. I don't have enough money to incorporate females in my research. Still not good enough. Females are too complicated. Definitely not good enough. So these excuses are no longer accepted, and the NIH wants to see females in your research. So females are not more variable than males. This is just one of my graphs from some research that we just actually collected like 2 weeks ago. I could give countless examples of this. I don't even know what estrus state these females are in. So a lot of people say that females are more variable because of the estrus cycle. So obviously females are cycling. We showed that graph of estrogen going up and down, progesterone going up and down. In mice, it's a 4-day cycle. But as you can see, these females, I don't even know what cycle they're in, but clearly my males are more variable than my females. It almost doesn't even matter what is on the X, my y-axis here. But this is just in a social behavioral task. So you see the mouse there. They have the choice to spend time with either a resident animal or an intruder or a familiar animal or a novel animal. And you can see that the males are sort of all over the place. The females are a little tighter. We have some mobility problems in the Flox Plus mice, which is interesting, but not the point of the graph. But we have tons of graphs like this from our lab, where we see just as much variability, usually more variability in the males than the females. So females are not necessarily more variable than males. And we will often we will almost always do a correlation with estrous cycle, and we usually don't see a correlation with estrous cycle. So in some cases you will, but that's an easy thing to do to incorporate in your research to control for those questions. So what can you do? So if you're interested in incorporating sex into your research, which I hope you guys all are, or already are doing so, what are some of the strategies that you can employ to get your grant applications funded, to have your papers reviewed favorably, or to just straight up interpret your data? I'm also going to toss in some things at the larger scale about how sex is perceived in the field, and to talk about some ways that can kind of, because all of this sort of comes together into one global perception of sex in research. So there's awareness, mentorship, and scholarship. And I'm going to go through each of these different bullets. Scholarship, of course, is probably largely why many of you are here, but I have the floor, and so I'm also going to talk about awareness and mentorship as ways to promote studying sex within research. So the first thing you can do is be aware of your resources. So NIH and the example candidate, Canadian Institute of Health Research, they have resources for you to access to see how are they interpreting sex differences. What are they interested in when they're reading grant applications? We don't talk about some of that, but they have some resources listed on their websites. Secondly, I already directed you to the OSSD and IGN societies, so those have a lot of resources as well. And even here within FBC, we have, of course, the Institute of Women's Health. And so they have a lot of resources available and a lot of great people that are available to talk about how to incorporate females in research with that group as well. Also, there's some joke videos. So I just wanted to pull one because I wanted to see if there was anything about cell culture studies, because that, of course, is a lot of people assume if they do cell culture that they can group all that together. But this is just one joke video I found, sex stratified neuronal cultures to study ischemic cell death pathways. So there's even some ways to incorporate sex differences when you're looking at looking at cell culture studies. You can increase your knowledge base regarding the influence of sex and gender and how it may be impacting your work and where the resources are to get. So this is a graph showing how the research group itself can impact attention to sex differences. And so in this graph, what you're seeing is whether or not the authors are attending to gender and sex analysis based on the composition of the author group. And so you'll see the estimated proportion of women within the author group leads to an increase in whether or not they are attending to gender and sex analysis. And so that is true at first authors, last authors, and the full. And so if you are working, within a diverse research team, you're more likely to attend to gender and sex analysis. So work in diverse teams, depending on what level you are, if you are the PI, if you're a postdoc, if you're a grad student, think about what your group is looking like and think about the diversity within that group and how you can promote that to attend to these details. And of course, we have a gender balance in journal functions. This is from the Journal of Neuropsychopharmacology. And what you're looking at here is a pie chart where corresponding authors are grouped as men and whether or not they suggest male reviewers or female reviewers. So if the corresponding authors are grouped as men, they only suggest female reviewers 25% of the time. If those corresponding authors are grouped as women, they still are only suggesting female reviewers 34%. Now, an easy question that is often asked at this point is about the field, the composition of the field itself. Are we a male-heavy field or a female-heavy field? We are a male-heavy field still, but how do we address that? How do we combat that? We suggest more female reviewers, because there are plenty of qualified female reviewers out there, to start to alleviate this gap, to kind of bring females and males as on equal standing with the field to help. Because if you're suggested as a reviewer, that's one of the ways you get promoted, that's one of the ways you become more well-known in the field. So suggest female reviewers. obviously don't sacrifice your science, but suggest female reviewers. Okay, so now for the strategies about actual research. So the NIH put out these extremely helpful and detailed guidelines for how to incorporate effects into your research. You're supposed to consider it, collect it, characterize it, and communicate it. Okay, well, communicate it, great. But what do you do about these others? How do you even begin to consider it when you never have? Or if all of a sudden you're doing a project where you suddenly have sex differences. And so I get this a lot where I have friends that will send me an email and say, I have sex differences in my recordings. I never looked at that before. What do I do now? Are my mice are a year old? Are they still ovulating? Yes, they're still ovulating. So when you never even thought about this, this can be a little bit of an overwhelming piece. And so we're going to talk a little bit about that today. So this is a really great place to start with your grant application. This is the rubric that the NIH gives to reviewers when they are reviewing your grant. And this is how the reviewer is supposed to interpret sex as a biological variable in the research. And you can follow this flow chart. And so basically it goes through and then you can address, if you're incorporating sex as a biological variable, that's a strength and you can address it. If you're not doing it, it's a weakness and you can address it. Just like a common question here is, how much do they care? Unfortunately, my answer is, it's hard to tell. It's individually based, as all grant reviews are, so there's always an element of luck involved in that situation. But by starting here, like I mentioned before with the definitions, you start with these definitions, you start with this rubric, you can at least get yourself into a competitive point. You can get yourself to where you know what you're talking about and you're able to incorporate this into the work that you're doing. I wish I could give you a better example or a better statement about how much they care, but I just can't. It also hasn't been that long that this has been a primary piece of these grant applications. So I think people are still getting used to it in the field and still kind of trying to get an idea of how much weight to give sex differences or sex as a biological variable in research. So that's about the best I can do on that, but at least starting here will give you a foundation to build on. So I like this picture because it shows you a bunch of different ways by which sex may be influencing your research. And so this is another good place to start if you kind of have no idea where sex might be influencing your experiments. This one is talking about some of the independent variables that we usually conveniently pretend don't exist. So things like the sex of your researcher or staff who's working with your animals. There is a number of papers showing that male researchers have different effects than female researchers on the behavioral outcomes of their animals. How's the temperature in the room? Anybody else a singer? How's the temperature? How's the sound? How's the lighting? What is the odor? I swear to God, I smell the food cards. So what are these different pieces and how might that be affecting your animals? And how do you have your animals grouped? Are they sex segregated are they male-female mixed, number of mixed animals in the lab? A lot of the times we have colony rooms where you're in a room with 5 other researchers or the researchers, and who knows what is going on with their animals, how they have housing. Do you have males and females? Yeah, okay, they're separated by cage, but are they right next to each other? Odor is their primary means of communication. They're smelling what's going on in the cages that are side by side. So these different pieces might be affecting your research and your outcomes, and sex is playing a big role in how these may be influenced. So strategies for incorporating sex as a biological variable at the social justice level and also at your research level. So sex and gender impact all levels of research and medicine. Awareness is the first step towards moving towards equity. And so when I mean equity, I mean bringing females up to the levels of males, both in research and in the laboratory. Attention to sex within our experimental design is essential. Diverse research and clinical teams improve the odds of equity. Diverse review teams, so suggesting more females as reviewers, is going to improve the odds of equity. And classroom and mentorship interactions are essential to raising awareness. Do you talk about sex differences? Do you talk about the sex of your mice with your undergrads that you're training in the lab? Do they even think about that as a piece of their works? So I have two very important slides that I want to kind of bring up. This one about some roadblocks to incorporating sex as a biological variable. So a lot of times people will say, okay, I don't have enough colony space, I don't have enough money, and females are very variable, but I'm going to do the best I can. So I have a sample size of 20 animals, and I'm going to now take 10 males and 10 females, and then I'm going to do the analysis by sex, and we're going to be good to go. Oh, look at that. My males and my females aren't different, so I don't have an effective sex. So I'm going to just pool everybody together and have that data presented as one group. So this woman, Dr. Ariane Albert, is a senior biostatistician. So she actually works for the Canadian Institute of Health. But she published this paper looking at a power analysis of sex differences and expected sex differences in research. So I think this is basically a pharmacology study for a lack of more needed context. But she's just looking at whether or not the female, like what is the expected effect? Do you expect that effect to be reversed in females? No effect in females, half an effect in females, 3/4 of an effect in females. And what you'll see is that if you expect very dramatic differences, it'll be easy to see at low sample sizes pure long data. So it'll be easy to see those differences and have a solid power for that statistical design. However, if you have a more subtle effect, which is almost always the case between males and females, it is going to be very hard to see that difference until you have a very large sample size. And so you all may have experienced this as well. I have many times now where more and more you're getting asked to do a power analysis. Reviewers are asking for it in publications. Reviewers are asking for it in grants. So do the power analysis. It's not that difficult. I'm happy to help anyone with power analyses if you need it. But by incorporating power analyses, you can better gauge whether or not you're going to see a sex difference with the number of animals that you're working with. And so then you don't fall into the trap of pooling together some groups that shouldn't necessarily be pooled together. Of course, on the flip side, sometimes it is straight up not appropriate to have sex as a biological variable. And this is an issue because NIH is trying to follow its guidelines. So this woman, Dr. Lisa Belia, she's from British Columbia in their Center for Brain Health. She is a personal idol of mine, also a scientific idol of mine, and she studies a lot of things that are female specific. So this is just a quote from this article she wrote for Vice. She got some reviewer comments on a grant where she had submitted the grant looking at the immune profile of the placenta. And they asked her why she wasn't incorporating males in that grant. Placenta. Males don't have a placenta. And so sometimes it's just not appropriate. And so don't feel like you are pushed into or forced into this situation that you really shouldn't be in. It's really a waste of time, energy, and money. If you're studying prostate cancer, you don't need to include females in that research. So if this is something that you are up against, depending on the reviewer comments, depending on how you write it up, you can address sex as an unnecessary, unneeded feature. You'd hope that it would be obvious, but even for someone as prestigious as Dr. Belia, that is not necessarily the case. So I do want to point out that sometimes sex is not appropriate, and that's okay. You don't feel forced into incorporating it into your research if it's not. Okay, so what can we do today? So what I would like you all to do is take a few minutes to think about how sex as a biological variable might be impacting your research. You can do this at whatever level you feel is appropriate. You can think about a recent experiment and how those data may have actually been influenced by sex when you maybe didn't think it was. Or you might think about how sex as a biological variable might fit into a future experiment. Maybe you came in here with the idea of, I know I really want to start incorporating females, but I don't even know where to begin. So take a few minutes to think about this and then turn to your neighbor and talk about those ideas, or you can turn to your neighbor right away if you'd like to just start chatting. And then once you guys have done that, we'll kind of talk about it out loud a little bit and maybe get some feedback. Any questions? Thank you. You guys want to come up? Everyone should out. I'm not reading. Because we want to see you. Hi. Oh, sorry, that's okay. You have no pressures. So do you incorporate this into
Actually,
are you thinking about it?
You know, right. We were grants for.
Yeah.
We didn't have.
So do you, so if you
mail along evidence. Intermediaries.
Yeah.
Yeah. There's a very.
Are you supposed to have? You make sure you're on the association. Did you already meet with them? Okay, you're probably already on the email list, so you should be good. Yeah. Cool. And so,
Yeah.
okay, so when you, I don't know, so you started the new lab. Okay. So our
First starting that my wife and I were taking the interaction with different stressors.
do you have to say no? Your hand?
I have never thought about this, but. Yeah.
Yeah, so I don't know if you, I mean, I have like your situation. I'm gonna try to think it's really interesting.
So we do some cultures primary.
Some of these sequencing stuff. Single cell. And so to me, it's like, I'm guessing you guys get all of your data from other labs.
Yeah.
Producing. I'm gonna consider it. That very well. Some figure out is where I'm telling you, I am back. So basically, so like, and so like if you don't get ungrouped by set, you don't really, you may actually be missing publishable. So I'm actually here to be I think that's what and I'm possibly collaborate or get career stability forever. I think more and more I'm also beginning to think about that. But
It's like my family.
This I did one and I did it for something else. They were like teaching. I don't have. Oh, good. Yeah, I got an idea. Oh, yeah, I was just I'm sure the house and I'm trying to figure out. I think you guys know I talked to you.
We do like the back to
And you are
right.
I'm gonna need to hear those papers in my interview. What are you writing? Yeah. About individual. I've worked with the so I'm having no share about that. I'm actually currently like my response factor is basically years. And so I was
Oh, gosh. I don't know, like,
but I know more than I would. It's like, I know other people have actually used Scrum, so we already do all of it. Should we be doing it better? What do we not do that I talked about? Just favorable. What? A lot of the short term oh, we don't. It's one of the main reasons we have to collections, but data analysis. I feel like I do we ever do shower by side? Oh, I do. We've done it all in the past. She goes, oh, Rachel told me, I don't know which one you're doing for everything now. Good job. Okay, so I think we'll come back together for the last like 10 minutes. Unless you have final thoughts, maybe 30 seconds for final thoughts. Great. Yeah, we need these things like a milestone. What? Oh. Okay, so any interesting ideas, thoughts?
I've got a question.
Yeah.
So, let's say you're running some endpoint and you get your results, you have a sex difference, but your variability is similar between sexes.
Mm-hmm.
Is it logical to move on to looking at the effect of cycle?
Absolutely.
Yeah,
Always. Yeah.
Okay.
Yeah. So if you have, even if you don't have variability, so say you don't have any variability at all, say your females are like crazy tight.
Mm-hmm.
What could be happening? Anybody?
So,
Why might variability be reduced? It could all be cycling together. It could be cycling together. So one of the things that happens in rodents in particular is they will start cycling together, especially if they're group housed. So you might have an effect without even knowing you have an effect. So whether or not you have variability, I always think tracking estrus, or at least taking uterine weights. And so a lot of times that's what we'll do, because it can be so mice you can track estrus just visually, and I'm happy to share we have very detailed protocols for mice and rats for tracking estrus cycle, and I'm happy to share those with anybody in the room that would like them. But mice you can track visually, rats you can't, so it can be a little bit invasive. So as you know, we're a stress lab, and so that can be stressful for the animals sometimes. And so we don't like to do it all the time. What we'll do is take uterine weights at the end. So when we collect the animals, we'll take the uterus out and weigh it, and you can get a gross estimation of whether they're either high estrogen or low estrogen, so you can at least get that much detail.
what if your endpoint was collected across like 5 days or something?
Oh yeah,
or something?
that's awesome. Then you hit every cycle stage. So if you're collecting, well, I guess, so it depends on if you're collecting. So some of our behavioral tasks will go for 8 days. So we know that, and we test every animal on every day. So we know that we're hitting all stages of the cycle. So every measurement we get is going to have encompassed all the stages of the cycle in those females. if you are, in some cases, and I'm thinking of our collaboration with you in particular, and in some cases you might be doing one animal on one day, and then another animal on another, and another animal on another, where you are kind of hitting multiple days. I would track cycles there, but also know that you're probably, again, as a group, you're probably hitting all the cycle stages. So I always think it's a good idea to have awareness of it, in case there is an effect there. But you can do it in such a way that, so a lot of the times what we'll do is we'll cycle our animals, we'll track their cycles for like the week before we start behavior. So we know like basically what they're gonna, because once you get the cycle pattern, you can be pretty confident in that pattern. So we'll usually kind of get it and then move forward, estimating what that pattern is. And then at the end of behavior, or like in your case, after you did tail withdrawal, then you just do a lavage and see what they were after you did the target end.
Okay.
Yeah, knowing when to do that can be kind of awkward because it can disrupt the animal some. So, anybody else? Anything else? So we were just talking about, Katarina and I were talking about, she's in the biostatistics department doing genetic sequencing. And I don't know how in tune, or this is something that our lab has been talking about a lot because we do some sequencing analyses. And more we're seeing that even single cell sequencing, people have statistical differences based on single cell analyses. And what a lot of times I was asking Karina, if people that give them their data, do they tell you what the sex of that sample is? And usually the answer is no. So another reason to incorporate sex differences in your work is there's probably publishable data sitting right in front of you, and you just don't do the analysis to see it. So it may be that if you incorporate that variable, you might be able to piece out some data that could be very beneficial for you getting publications. And of course, that's the currency of science. So it may be that just by analyzing some data, already existing data, just analyzing it by sex, or in the case of these genetic sequencing studies, you very likely have some differences just waiting to be published. If you're advancing your postdoc or actually at any stage, that's what you want. So. Anybody else? Is share what you're talking about? Oh.
We were talking about in the same program.
Yeah, and I'm in bioinformatics,
Oh,
but I came from a cortex research lab. So we did behavioral studies, but since we were contracted, everything was only male, long Evans, and it's model for relapse. So very strict. But it would have been really neat to actually see differences in sexes with females and seeing if they relapsed either quickly or actually.
Well, so if you guys were contracted, that means you're probably doing toxicity or efficacy for a drug company.
Mm-hmm.
So is that drug going to go on the market? Are females going to be taking that drug?
That would have been great to know, but
yeah. And at the clinical trial level, you're looking at you walk into clinical trials and all of the existing data is in males, and all of a sudden now you're at human women. So you basically just went from 0 to 100, and then you're now going to be dosing human women with something that's how it could go. And of course, the opiate graph that I showed before, there's significant sex differences in opiate responsivity. So.
Anecdote data, I saw the news. I didn't get a chance to read the article, but some new treatment for HIV came out, I think today.
Oh, shoot, yeah.
But it's only approved by the FDA and because it's stuff that's tested in females. So, it's cool.
So of course, when you get to a situation like HIV, cancer, any of these diseases that are high risk, high mortality, one of the problems you can come up against is women might see, okay, this guy I know is taking this drug, like this is amazing. I want to take this drug. I want to get in on this. And a lot of the times there's difficulty in how the drugs are marketed and how they're presented for patient intake and how those patients perceive that. And if there's not, because you can imagine that, someone from the average population, they may not be aware enough to consider is this safe in women? Like you're just talking about this drug, or how this person taking it. I want to take this drug. So you already have some concerns if it's not done that way. Cool. Any other questions, final thoughts?
Do I have anyone sorry?
Well, thank you guys very much for coming. Thanks for your attention. Thanks for engaging. And I'm happy to provide any assistance if anyone ever needs it. Our lab does this often. So.